probably start from the cell cycle stage itself, so that it would be a good recap. So here we are. So I'll do this. Fine. So is it visible? The slides?
Yes, sir.
Great. Thank you.
Yes, sir.
Yes, sir.
Thanks. So
Yes.
we learned that for a faster cell division, bacteria has to produce more of their kind. Um then that's why they started with the most simplest way of cell division. Here, the checkpoints or checking in the correction in the DNA copy hardly matters, because see, if a cell is damaged, individual isn't damaged, so the cell will die. Already that the cells are unicellular in nature, so not a problem. Their whole genetic line cannot be damaged. So if a bad cell generated, so that cell will end up in a bad cells altogether. Its generations to come will be bad, and finally it would be abolished. There is risk in multicellular organism. If there is a bad cell somewhere, then there is high chances that that can cause death of the whole individual. Okay, so that's why eukaryotic cells, which are specifically multicellular in nature, they do not do binary fission. They have a lot of checkpoints, proofreadings for the DNA, and all. So what do you call the cell divisions of which are very symmetric, very corrected cell divisions of eukaryotic cells? You call that mitosis. Okay, that's a process. That's a cell division process which duplicates cells in eukaryotes. So what are the stages? Prophase, metaphase, anaphase, telophase, cytokinesis, and there is one predecessor phase, that is interphase. Similarly, animal cells also, here it is. But here the interesting thing you will see, animal cells, the centriole, this is particular organ you will see additionally in the cell. In this case, in plant cells, you will not see any centriole existing. This is the picture, it's not a drawing. In drawings also, they will not show you any centriole. As we understood that the polarity already exists in plant cells, rigid cell wall gives a polarity, and in animal cells, as it is very, you can see very irregular in shape, the centriole makes a particular spindle fiber cage, and that gives a proper polarity. And you see this similar anaphase, and cytokinesis there. So that means the cytoskeleton, which is often made up of microtubules, and there is microfilaments. They work a lot during the cell division process. So we learned there are what are the fates of different organelles in cell during cell divisions. Now, when we see that cell division happens, and this is the original size of the cell, and suppose this is the original size of the cells. You see, the cell is so big, and at the end of the cell division, you see there are two smaller cells which has been generated. This cell has to come back to the same size which the mother cell which has started cell dividing. So that process, preparation for the cell division, that process is called the interphase process. And often this interphase process, when cells don't want to divide at all, there is additional phase called the resting phase, quiescent stage. We can call it as G0 phase. So interphase is a preparatory phase for the cell division, but G0 phase is a resting phase for the cell division. From G0 phase, if the cell all of a sudden thinks that I can divide, then it has to cross a checkpoint, and go through the G1 phase, then S phase, and G2 phase. That's how it is. And then after G2, it can finish the mitotic divisions. Mitotic phases, we have learned that prophase, metaphase, anaphase, telophase, cytokinesis, right? And you see one cell, another cell is duplicated, and the original cell will again go into the cell cycle. This cell will again go into its own kind of cell cycle. Okay, it's not a particular physical cycle. We are showing it. It's a stages of cell. That is how we are calling it a cell cycle. And what are the checkpoints we see? MPF and cyclin. If you damage these checkpoints, there is high chances that cell will end up being cancerous. Okay, so we saw this thing also, how cyclin regulates, how MPF initiates the mitosis process, M phase, that is mitotic division process. Now, what happens in the synthetic phase? That's what we were discussing. G1 and synthetic phase. S phase is significantly dedicated for DNA synthesis. So one of the thing which happens in the DNA synthesis phase, and of course the subsequent, G2 phase, not G1 phase, is that DNA is replicated in the S phase. It's a lot of DNAs chains are there. Almost 23 DNA chains, 46 DNA chains are there. That's why 46 chromosomes. That is 23 pairs of chromosomes. At least in human being, I'm saying. Many eukaryotes have different numbers of chromosomes, and that's very characteristic of their species. So these DNAs are replicated. So when you define life, there should be a molecule or some basis where the molecular information, the constructional information of the cell can be recorded and can be passed through another generation. In Earth kind of living system, we have DNA which can have the memory, or which can have the informations how the a new cell will be constructed back. Then if this is so, can I just simply put the DNA and ask it to replicate? Can it produce a cell? No. So DNA is the central molecule, then it needs support from other molecules too. So you simply put it together will not work, give you a working cell. You need to put things in a particular place. That's how an industry or factory also will be. You just simply dump all the machines on a place. Will a factory stand there? No. So here we saw that DNA molecule is there. You can see two helix of the DNA molecule with two spiral setups. That two spiral, that's why we call it double helical DNA. Recently, it had been discovered that in some positions, this DNA is 4 helix, quadrihelical structures in some places, human DNA, that we did not knew earlier. Now we are able to understand. Now see, this is the DNA. DNA has to be tugged up properly, tied up properly, so that no mixing happens between DNA molecules, and it should not tangle back. It should be able to open it. Such long thread, and it never tangles. That's very surprising. So here it is being compacted on and on and on and on. Finally, it makes a chromosome like this. Okay, so what is the diameter of the DNA molecule? It's somewhere 3 to 4 nanometer. Okay, and now you see the chromosomes are somewhere multiple micrometer thick. So this is what the difference happens. So multiple DNA chains tugged with proteins, and now you get chromosomes like this. Okay, this is what cell divisions are all about. And any eukaryotic cells, be it single-celled or if they are multicellular organisms, they divide through this mechanism. Okay, now in humans, let's talk about multicellular organisms. What are the kinds of cells you see? In humans itself, you see there are almost 32 trillion cells in an adult human being. And these 32 trillion cells actually are not all the same. You can see in this picture, they look very much different. You know? So how many types of cells you can see in human beings? You can see 200 types of cells in human beings here. Okay, 200, it's not a small number, okay? And every day you lose a billion cells. 10 to the 9 cells. Every day you lose, and cells are replaced by newly growing cells. Okay, so that means a human being, even if it is not growing in height, still needs cell division in its body, which generates to generate at least a billion cells, which can replenish the old dying cells of your major places where which shades the skin is your mouth, your intestinal canal epithelium, your skin. These are the places where you majorly shade cells. Now, in case of plant, a single cell seed or zygote, from that you see different kind of cells. These are mesophilic cells, these are this xylem cells, pink where these are companion cells. These are again another mesophilic cells. So see, there are really different kinds of cells in a multicellular organism. Now, my question here is that, and we all know that when they become a multicellular organism, just take an example of human being. Does it happen that heart demands, because heart works 24/7, heart demands special rights or goes into strike for some time? Does it happen? Or does heart cell invade your liver cells because so we are working very hard, we should need more food, I need to invade liver? Do they do that? Similar thing happens in India, right? And in societies. And if you say the cellular systems in our body are societies, small societies, well, the rules are different, bit of different. But yes, cooperative action is there. Division of labor is there. But can we say that this thing can happen, invade cells from the organ, one organ to another organ? Does it happen? This is a question to all of you, I guess. No, it never happens. Even if it happens only in disease condition, and that individual will not survive. This happens because when you have cancer in liver, this cell will not stay contained in liver. It says that no, I don't want to stay in liver all along my life. I want to visit all through the body, and I have to get friends from that places also. It will visit the lungs, it will visit the kidney, it will get friends from there. They will make another gang, they will again visit to other organs. And that way, when cancer patients die, they do not die out of some infection. They die out of organ failures because these rowdy cells teaches other obedient cells to stop harmony. And people die out of multiple organ failures when they die out of cancer. So that means cancer is a case where the total collapse of the social network of the cells, what are the rules they are governed with, they're there. This happens. That means during the development of the first, multicellular organism, why did they took so much amount of risk to stay together? And you can say that at any moment you can have when there wasn't proper immune cells, who was catching this kind of cancerous activity? Nothing. So still there is high chances that the overall system gets disrupted. But you see, although the numbers are less, but you see a lot of multicellular organisms in the on the face of Earth. My question is that why, if multicellularity has happened, I just need to know what must be there in the mind of God or the nature, which has simply preferred multicellular organisms to form. It's not the most preferable form, although. Yeah, still we have individually staying cells more in the face of Earth. Their biomass is also more. But still we have multicellular organisms. Why do they take risk of having multicellular organisms?
So I think you can look at it this way. Even humans survived over other species. Even humans because we live in society, species right? Because we live in society, boil it down to cells also. Multicellular organisms can show the cells also. Multicellular, even if some cells are lost, they can compensate with other cells. Even if some cells are lost, they can also have specialized cells performing specific functions. You can also have specialized cells performing adaptiveness.
Can you say really it's a very unique thing to, because see, the most favorable thing is still your individually living cells. You know, most number of cells or living beings present on Earth are single cell organisms. Yeah, basically
Yes.
bacteria and protists. So
Yes.
so first we took a risk of making a eukaryotic cells, then we start again making more complexities, then we made cells to stay together. And yes, we see a beautiful thing, beautiful way of coordination. And so when you see a juggler, you know, throw their catch it, you like it very well. When you see a baby throws its ball, and you may not like it, right? And normally you won't like it because he's failing all the time. But how many of us can become a juggler? How many of us can become Virat Kohli or Tendulkar? Majority of us, we can simply hit our bed and don't know where the ball will go. Of course, a symmetry, a synchronization we all love. Does nature have this special love for that, or it had got a special scientific logic to that?
So I think the one way of looking at it is,
Yes.
the one way of looking at it is you go towards you always try to go to a local minimum. Evolution tries to take you to a local minimum. So unicellular organisms found a way to travel a path and reach one local minimum where they could, I mean, path and reach one local minimum. They could, when a slightly different evolution happens, for example, a slight different evolution happens, for example, they couldn't sustain themselves. Another evolution would have taken place whereby you can have
When you say
you
local
can have
minima, what
cells
minimization they are
together and
doing? What minima they are looking for?
an optimal solution rather to survive that condition. An optimal solution rather to survive that
Um,
conditions.
no, that is not a case. See, in all, as I said, in all living environment, you know, in Earth we have different environments. Okay, in all living environments, you will not see eukaryotes or complex living cells, living organisms are existing. But in all living environments, you see individual single cells are there.
Yes, sir. Why should we look at it in a linear way? Why can't we look at it
Why should we
like it's a
look at it in a
branch?
linear way?
Why can't we
Why
look at
should
it
we look at?
like
My
a
point is that
branch?
why nature had looked at it in the first place? That was the question. Let's try to answer. Like, yeah, there is something minimization involved here. I'll just try to come to that. Whether I don't know. These are the certain answers I have, and some scientists also have it. But is it the final answer? We don't know. Okay, let's see. This is how multicellularity happens. You see, you have this hydrogen atom, oxygen. This is water, how it makes, and you know this water molecule, and multiple molecules make cells. I'll go deeper into that. And these cells, this is a smooth muscle cells. Multiple of the smooth muscle cells makes a smooth muscle, which can work actually. Okay. level organization. This is atomic level organization, molecular level organization. There is in between supramolecular organization. When you see this microtubule, multiple different molecules of microtubules, they are coming together to make the microtubule. This is a supramolecular interaction, and that is the basic, cue of Lego story of life actually. Yes, from there we get functional living cells. Living cells becomes tissues. Tissues becomes organ. Now your bladder, which where you store urine, all of us have this. It is the wall of the bladder is lined with this muscular smooth muscle cells, and now you have this single organ developed. The single organ gets fitted into other organs and works in coordination with them. Okay, so when we see this multiple organs works in coordination with each other or optimal function they achieve, we call it as an organ system. Usually is created like when multiple organ systems work together, we have an organism happening. Now, the question what we were discussing, that why from this one we at all went to this one. Did nature envisioned human being produced after 3.8 billion years, or is there something very quick target or quickly achievable when they are clubbing multiple cells together? That is the question. Suraj tried to answer. There is some sort of minimization thing, local minimization they are looking for. Yes, probably there is two varieties of minimization they are looking for. There is when now, if you have a human being of 6 feet, okay. The question we are discussing, why we do not have a whole human being of a single cell. Okay, we have amoeba. Amoeba can walk. Amoeba can have senses. Amoeba can hunt. It's a single cell. Okay, and why we have to have this 32 trillion cells in our body? Okay, so here is the first most basic geometrical answer to it, that a cell has to sustain most in the best condition if its surface to volume ratio is minimum, and that's why majorly you see cells have a spherical shape. Sphere has the least volume, surface to volume ratio. It has the least surface to volume ratio. You see here in these three things, here you have a small object, okay, small cube. Here you have a large human-shaped cube, and here you have same cube, but which is made up of small modules of this size. Which has the minimum surface to volume ratio? Of course, the large cube, when it is, although the smaller cube has the minimum surface to volume ratio, but when they make a larger cube also, these small modules, the surface to volume ratio did not compromise. So if that happens, what is the benefit cells get? So we understood that the basic unit of living systems are cells. When you club multiple cells together, or if you make a large cell, there is high chance that for a small glucose molecule to reach, get converted to an ATP and reach your nucleus may take a day or two if the whole cell is a human-sized cell. How fast do you need an ATP to reach to your nucleus if they are actively dividing DNA? You need it very instantaneously, very fast. Okay, that is why you need to have. See, cells do not communicate with electromagnetic radiations. Okay, they in the cell things has to function with mobility and transport of different molecules. And how this mobility and transport of the molecule can be most feasible, most efficient, if individual cells have their own local centers and they are functioning individually. Cool. So that is the reason we need a smaller surface to volume ratio. Yes, but why we need to suppose we started making a multicellular organism. Multicellular organisms may have lesser, but why at all make it? We saw that surface to volume ratio is of the smallest cube here is the same as that of the large cube. Why a large cell isn't there and small individual of multiple cell is there? It is, we understand. Why at all a large individual is being formed? That question isn't answered. That question answers the second point, energy minimization. So there had been a lot of studies on multicellularity, development of multicellularity. We all know that living systems, they need energy to sustain, right? Energy in the form of food, okay, and they do their metabolic energy production from the food, and they run their chemical factories and systems, right? Now, this is a very specific answer here. When you make the size of mass of an individual double, okay, if it is X and suppose if an individual is 100% of its size and you have made an organism of 200% size, suppose evolution made the organism of 200% size, what you can say, like me and a cow probably, okay, I am 80 kg, and if you say a very grown up cow or a Royal Bengal tigers can be of 200 kg weight. So here, when the mass doubles, is a number of cells, everything we think that doubles, that doubles. But one surprising thing happens here. The energy consumption of reduces by 25%. Understood? So now I am consuming suppose 100% energy for 100% mass, but for my 200% mass I will consume only 150% of energy what I used to consume. Is it fascinating? When there is competition, and see, we all knew that there has to be individual single cell animals or organisms. From there only they started collaborating and started producing multicellular organisms. But what is the perk in here? The only perk in here, why joint family still exists, they, with a single cook, you can feed 10 people. If you go to hotel with a single buy, can you feed 10 people? No. You cannot even, the hotel person, the hotel owner may not allow the second person to eat in your plate, even if you leave the food away. This is the energy minimization idea, and actually when they started studying in single organisms, when initial multicellular organisms starting to be built, they found that this energy minimization was one of the biggest perk which was being worked on, and that's how multicellular organisms were developed. Now there is again a question that why everywhere they do not have multicellular organisms? Why the number of multicellular organisms aren't more? The answer is everywhere they did not have that rigorous competition. That's why multicellular organism wasn't there. In some places where you see, you can see skyscrapers in Bombay and probably in Delhi, 80, 70-story buildings. Do you see those skyscrapers in Vellore? In Vellore, we have only 12. The tallest building we have in VIT only. In local areas, we may not see, small towns, we do not see these skyscrapers. Only some cities you see skyscrapers. Where there is space available, why to make skyscrapers, right? Okay. So that was one of the story here, the reason why multicellularity exists. And to understand the level of organism, we need to understand the scale, length scale. For understanding the length scale, we need to understand this here. Okay, so just see that atoms are of 10 to the minus 12 and gradually organisms are of. I'll just bring you to a web page. It's a very, I think I shared the link also. You can what is this? Okay. So it's a very interesting page. You all have printers at your home, or you know printed letters, and the printer will have a default printer setting. Now it is Calibri, but earlier MS have Times New Roman as a default letter, and its size was 12 font. So a 12 font letter will be bit larger than that of a sesame seed, smaller than of a grain of rice, and of course a lot smaller than a grain of or a coffee bean. Now, and you can see the scale here, millimeter cross millimeter. Now we understood this is the organization, level of organization. Now let's understand the scale of organization. This is rice sesame seed. Now let's go deeper. Okay, now this is a slider. We can go deeper. Okay, this is just diving into the smaller things. Now we have 100 units of 1 mm. Now you can see a grain of salt here. Then you can see a multicellular organism, amoeba here. And sorry, single unicellular organism, amoeba. Amoeba is larger than paramecium. This is again a protist unicellular organism, single-celled. Don't make your skin cells, which you shade every day during brushing and that is again 30 micron. Your hair diameter is 30 micron in when I say micron, is it's 1,000 times smaller than a millimeter. So often people called it as millimeter. You can say that. Okay. So micrometer, that's what micrometer is. So in micrometer scale, you see majority of the cells you can see. This is the photo. Rod-shaped thing is a photoreceptor of your eye. You see, it's 100 micron long and 2.5 micron wide, micrometer wide. You have your sperm cells, and this large ball-like thing, it's an egg cell. What is the size of an egg cell? It's 130 micrometer, the diameter. Sperms could be 60 micron in length and 5 micron in diameter. Then comes your red blood cells, that is 8 microns. It's very small, you see. And the reason you see red blood cells can't have chromosomes, it's only one chromosome has a length of 7 micron. It cannot fit in the red blood cell. Can you see that? And then your baker's yeast, you eat it in your breads and cakes. Then comes to E. coli and your organelles. So as you see, E. coli's length and your mitochondrial length is almost similar. The dimensions are similar. So that's one of the reasons we think that mitochondria must have been a prokaryote earlier, and it has entered into the eukaryotic cells, and that's an endosymbiotic relationship we carry. And we have the small organelles, lysosomes, and now we come to viruses. Okay, measles virus, then influenza virus. Coronavirus is somewhere close to this measles virus. So micrometer is gone now. We are diving into nanometer scale. That is another 1,000 times smaller than your so that is again 1,000 times smaller than your micrometer. So you see in the nanometer scale, you see majority of the viruses, and when you see a bacterial wall, cell wall, and when you see a virus, you can see viruses so tiny. Okay, so this is a bacteriophage, which this is specifically some viruses which infects bacteria. You know, these viruses infect human beings. These viruses infect this is a mitochondria, but its dimension is similar to that of a bacteria, normal bacteria. Now we go deeper. The smallest virus, you can see the hepatitis virus, and we can go to no, smallest is the rhinovirus, sorry. And then we go deeper into the smallest organelle. This is the smallest organelle. You see ribosome, it's 30 nanometer wide, but one of the heaviest, densest and heaviest organelle, which is majorly the machines which make proteins here. Now, when you see here is molecules like hemoglobin, which majorly is present in your red blood cells, then your defense molecules, this is antibody and RNA molecules. These are 7 to 12 nanometers length. Methionine is the constituent of your protein, individual protein. This is a small amino acid. Adenine is a constituent of RNA or DNA. Then you come to a picometer scale where molecules are there. Glucose, water molecule, and comes to carbon molecule, carbon atom itself. It is 340 picometer. This is the length scale. Now, from the carbon atom, we will slowly go up. We'll go back to the copy page. So pico we left, now we are in the nano realm, and then we have got the micron realm, then this micron realm going on, we go to the millimeter realm, and we are back from where we started. So this is the length scale. Now, when we are talking about tissues, this grain of rice, sesame, the tissues are of these ranges, millimeter in size. Okay, when multiple cells meet together, an organ can be some more millimeter in sizes. That means your heart can be of 100 millimeters height, and it can be width. It can have a width of 15 millimeter, 150 millimeter. That's all. Organ systems have a centimeter or centimeter scale. Humans we can define in meters. Okay. So this is the definition also: meter, centimeter, millimeter, micrometer, nanometer, angstrom, and picometer. Angstrom we all know. Angstrom is 1/10 of a nanometer. Okay. So today we discussed about different, length scale and organizational levels. So next class we will try to wrap up the cell biology part, and we'll move ahead. Okay. So if there is any query, you can. Yes, any query you can ask me. Okay.